Okay, so now we're starting on the part of the class which I call gene expression and disease, where we try to explain the genetic basis for various diseases as a result of errors that have happened, of course, in the DNA, but which end up showing up as in the phenotype as a protein or RNA that cannot perform its function in the cell. So I'm starting at the end, really, which is a mature mRNA, which is ready to be translated to form protein. Okay, so producing this mRNA involves, of course, a lot of steps, including transcription and then for the protein translation. And all those processes need to go correctly in order for the actual mature mRNA to function properly. Okay, so the parts of the mature mRNA from the beginning are the 5' cap, and I'll say a little bit more about that in a bit. But then there's other parts of it that people tend to forget about, which is that not the entire mature mRNA actually contains codons that are translated. So the 5' UTR is also called the 5' untranslated region or leader. Okay, this is the part of the mRNA which is before the open reading frame. And the nucleotides here don't count as codons because they're not translated and they don't contribute to the sequence of the protein. This doesn't say they have no function, but in looking at the actual protein sequence, which is going to be made from this mRNA, those 5' UTR nucleotides just don't count. Next, we have the coding sequence, or ORF, open reading frame. This will start with an AUG start codon and end with the first in-frame codon, stop codon. Okay, we'll talk about that again a little bit more, but remember that the ORF is made of 3 nucleotide-long sense codons that specify amino acids, and we read those, of course, from the genetic code. Okay, so we're looking at the coding regions still. Remember that eukaryotic genes have introns and exons. So the mature mRNA is made up of the exons. The introns have been removed. So splicing has occurred, and this has to occur correctly for the exons to be put together properly so that the protein can be read as needed. After that, we have the 3' UTR, or untranslated region, or trailer. And this part is after the stop codon. And again, like the 5' UTR, it's not translated, and the sequence here does not form part of the protein. Seems like a waste because sometimes there are a lot of nucleotides there, but they can be very important, actually. Finally, there's a poly A tail, which is very unusual in that it is not read from the DNA sequence. It is added to the mRNA or pre-mRNA as part of the processing. Okay, so the theoretical example in our textbook is missing some kind of reality to it. So I want to show a real example of a beta globin mRNA, and I'm going to show you this entire thing as a worksheet, and we're going to be working with this a little bit later. So what we're going to see is what's called a CDNA, and it's going to look a little startling to you because we're not having what we think we should have in an RNA because it's made of T's rather than U's. So this is clearly a DNA sequence, not an RNA sequence. And the reason for the T's instead of U's is that this particular molecule was created in vitro by copying an RNA template to eventually form a double-stranded molecule. So the molecule we're showing here we're only showing one of the strands because that's the strand that we can actually read to show us what the protein sequence is supposed to be. It also has some little things in here for our own convenience. Notice that the nucleotides are separated, grouped into groups of 10. There's nothing biological about the groups of 10. Those are for our own convenience in looking at it, making it easier to see, and being able to talk about specific nucleotides. Okay, the nucleotides are also numbered, starting at the left with number 1. 1 represents the first nucleotide of the RNA, which is to say the 5' end, and then it continues reading line by line. We're not seeing both strands, so we just read line by line until we get to the stop codon, which is located further down at nucleotide 494, I believe. Following that, we have the 3' untranslated region. So it doesn't look like an RNA sequence. It's not. It's made from one, but it gives us a way to look at them and talk about them in ways that can make sense to us. Okay, remember it's 5' to 3' on the RNA, which is to say the cDNA is also read 5' to 3'. Okay, it's grouped to 10, etc. Okay, if we look at this, we have to say, okay, how long is the 5' UTR of this CDNA, and how long is the 3' UTR? Okay, so if you look at the sequence, I made things easier for you by putting little marks on the AUG and the stop codon, which is UAA or TAA. So the AUG is actually located at nucleotide 51 in the cDNA. So that's where the coding region starts. The first 50 nucleotides are untranslated, and those are not part of the protein coding region. Okay, the 3' region, the 3' UTR, occurs after the stop codon. Okay, and that's really quite a long sequence that's there after the UAA, TAA stop codon, which ends at nucleotide 494. So I get 156 for this. Some people include the stop codon, and some people don't. It's okay either way. Okay, let's look for a minute at the AUG or the ATG, which sets the frame for translation. And we're going to come back to this idea in a bit, or actually at the end of the class. And I want to just point out now that you can tell what the amino acids are going to be in this protein by starting at the ATG and then yourself breaking them off into groups of 3 to see what the actual codons being read are going to be. So we're going to look at the next nucleotide, which is GTG, then we have CAC, etc. And we can go to our genetic code and then look and see what that GTG encodes and then read that off the chart and write it. So in reading these things, that's the first thing we do is break off groups of 3, starting with the ATG, and then go to the genetic code page and then read what that actually says. Okay, by the way, the first GAG is the glutamic E. That's the one that's mutated in the sickle cell allele of this gene to give GTG encoding Bailey. Okay, so here's the genetic code in our DNA form, and we will see it sometimes in the RNA form or the DNA form, depending. The cDNA is in the DNA form, and so we're going to use that to make it easier to read. Okay, so the first way to think about this is to see how the chart itself is organized, which is not obvious unless you look at it. Okay, so the rows, T, C, A, G, reading down, tell you what the first nucleotide of the codon is. So everybody in that row has its codon starting with a T. Then we look at the columns, and that tells us what the second nucleotide is. So if you start with the top, read your way down, the second nucleotide is T down the row, down the column rather. And then, of course, the last nucleotide in the codon is within the little box, just to make it easier to orient. Okay, so we've got 64 total codons. 61 of those are sense codons, meaning that they encode a specific amino acid. And there are 3 stop codons, which are sometimes called stop, sometimes called TER, as in this chart, and also sometimes indicated with an asterisk. There are different ways of representing the stop codon. Okay, notice also in this particular chart, we're told which amino acids go with which. For example, the first nucleotide, the first codon there is TTT, encoding phi-phenylalanine. That's the 3-letter code, which is in the single letter code F. So each amino acid has a single letter that designates it, which you see most of the time because it's easier on the computer. But each one also has a 3-letter code, which is older but a little more intuitive because it kind of tells you what the amino acid is. Okay, so notice about it too, for example, some of the things make sense, like leucine is L. Fine, L is leucine. But that leaves us with lysine. Well, L is already taken, so lysine is a K for some reason. Okay, also notice where there are little marks below, like for example, the TCT, TCC, TCA, TCG, all encode serine. So that's indicated rather than writing serine 4 times. They just put little marks. Okay, here's your RNA code, same idea. This is a little bit clearer, I think. Okay, I think I probably said all this. Oh, yeah. Okay, so some amino acids are encoded by several codons. This is called redundancy, right? For example, there's 6 leucine codons, but not all amino acids are encoded by more than one. The AUG is unique for methionine, and the tryptophan is only TGG or UGG. But we still read in groups of 3. You should practice reading these and practice being able to work on this chart. Okay, I'm mentioning the stop codons now just to keep in mind that they're going to become important later. The open reading frame ends when the first in-frame stop codon is encountered. Okay, so in this case, it's nucleotides 492 to 494, and I'm showing it this way as an asterisk, right? So now that we know what we're supposed to produce from our genes so that a protein can be made, I want to go to the beginning or the back up to talk about the DNA and talk about what the gene is like and how it goes through all these processes to create the mature mRNA that we're going to translate with our genetic code. Okay, so remember a gene occupies a particular location on a particular chromosome, and not shown in our mature mRNA or even our pre-mRNA is that there are regulatory regions, which are in addition to the transcribed regions. So it's important to have part of a gene before the part that encodes the mRNA, because that's going to do things like cause RNA polymerase to bind and transcription to be initiated. Okay, so once you've got your DNA ready to be transcribed, RNA polymerase will transcribe it to produce a pre-mRNA, and that needs to be processed by removing introns, putting on your cap, etc. And then after that, it can go to the cytoplasm for translation and then create the protein. So there's a lot that has to happen correctly before we can actually produce a protein from this gene. Okay, so reiterating really, the information within the DNA sequence is in some sense converted to a protein sequence eventually by transcription to produce the RNA, right? The RNA is produced by the process of transcription, processing, polyadenylation, capping, and then translation. Okay, so the idea is that any change in the DNA sequence can lead to changes in the protein sequence as a result of mistakes in almost any one of these processes. And as a result of this kind of misfunction, biochemical pathways and other pathways, processes will go wrong, and that can produce disease. Okay. Again, we will review these processes very quickly and very briefly, and then think about how changes in the DNA sequence, which is to say mutations, can cause them to go wrong. Mutations in any of these steps can lead to disease. Okay, so you can see the blue thing at the upper left-hand corner designated as the words promoter. Okay, that's the part of the DNA sequence which is essential for transcription to occur, but it's not itself created an RNA. There's no RNA, well, let me say there's no RNA. The RNA from this gene is not made from that part of the gene. That part is only to get transcription to start in the correct location and at the correct time. So following that, we will have, of course, transcription, which produces a pre-mRNA that will need to be processed to produce the mature mRNA, which will then be translated to form the protein. So this is a slightly different representation of the mature mRNA at the bottom here, where they're very clearly showing you the cap, the 5' untranslated region, the 3' untranslated region, and the poly A tail. So that's the molecule that's presented to the ribosome for translation purposes. Okay, so it's important to think about signals in the DNA and RNA to cause their correct function. Binding of proteins to nucleic acids is a very important part of this whole process, and it's kind of hard to imagine how a protein can recognize a specific DNA or RNA sequence, but recognize them they do, and that's a very important part of proteins that are involved in transcription, translation, etc. Okay. All right, so processes and signals for them. Transcription by RNA polymerase to produce the RNA. There are signals that are in the gene that are required to bind proteins to get polymerase to the DNA to get the actual transcription to actually start. Now, these vary a lot from gene to gene, and so there's no rule about all genes must have such and such. So there's a lot of variation among genes as in terms of what those sequences actually are. So here is a minimal eukaryotic upstream region. It includes a sequence called the Tata box, which is there in many genes, but by no means all. Then in addition to and that occurs within a short distance before the mRNA initiation or the first nucleotide of the mRNA is going to be copied. Then we have, in addition, upstream promoter elements, sometimes called proximal promoter elements, which are located before or say upstream of the Tata box, and what they do is bind other proteins, which also contribute to transcriptional enhancement or repression in some cases. Okay, so these proximal promoter elements include short sequences such as GC box, cat box, oct box, and others. And these are named according to what their sequences kind of look like. For example, the cat box binds the cat box factor. Okay, fine. Okay, so here are a few examples of eukaryotic genes showing the location of these various sequence elements that occur before the transcription start, which contribute to the expression and regulation of these different genes. So as we see, a lot of them have Tata boxes, but the SV40 virus early promoter, for example, does not. GC boxes don't occur within the beta globin proximal promoter region, but they do occur in the SV40 in many, many copies. Well, 1, 2, 3, 4, 5, 6 copies, etc. So some genes have cat boxes, oct boxes, whatever, whereas others do not. There's a lot of variation in that. Okay, it's also the case that sequences outside the proximal promoter region can also regulate transcription, either in a positive or negative manner. Okay, enhancers have much more complex sequences and can be very far away and still control transcription. Here's a picture from the Cell and Molecular book showing this. We have the RNA polymerase, the blue thing, sitting down at the promoter. We have a bunch of transcription factors which have assembled at the Tata box. And then we have a little blue thing, which would be an example of an upstream promoter element. And then we've got all these sequences further away, which have gotten to the promoter or near the promoter by means of some process where the DNA is able to, the proteins bound in the DNA are able to interact with proteins bound at the promoter, even though they're very far away. Okay, so once transcription gets going, it moves along the template DNA, copying one strand as it goes. Right, the new molecule is extended in the 5' to 3' direction as it goes. The template, of course, is complementary to the new mRNA. The best way to think about this is that the first nucleotide of the mRNA will be the 5' end of that mRNA, and it's extended in a 5' to 3' direction on itself as transcription occurs. Okay, so here's our picture again. What we've done is made a pre-mRNA by copying the DNA template, and that's in the second line of the picture, starting with the first exon, which is going to be included within the mature RNA. Then we have 2 introns, which are going to have to be removed. The exons are shown in green, the introns in orange. And then we have some downstream sequences where we need some things to happen, as well as the 5' end capping. So we need to do quite a lot to this to get it to the point where it's going to get translated. Okay, meanwhile, here's a question. A particular gene is missing more than 1,000 base pairs from its upstream region, including the Tata box and proximal promoter elements. As a result of this mutation, the pre-mRNA will not be produced, the protein will not be produced, RNA polymerase will not bind, all of the above. Okay, if you said all of the above, you're smart. Okay, good. A mutation that removes the proximal promoter elements from a gene would likely lead to reduced transcription, reduced production of the normal protein, production of an altered mutant protein. A and B are both true. A, B, and C are both true. I would vote for D in this case. So now that we've got our primary transcript, we've left behind the promoter and other upstream sequences, but we have our pre-mRNA now, which needs to be processed. And it needs to have its introns removed. It has to create the mature mRNA, which is what can get translated. Okay, so the 3 pre-mRNA processes we need to at least mention are capping, polyadenylation, and splicing. All right, so there are signals for all these processes, just as other DNA or RNA-dependent processes are, that there's specific sequences to which proteins bind to carry out those functions. Okay, for capping, the signal is actually just the 5' triphosphate, right? Polyadenylation by RNA polymerase has a signal, and splicing by the spliceosome also has signals that dictate where that splicing should occur. Okay, so I don't care for you to learn a lot about capping. I'm going to put it here for the purpose of people that are going to have to take the MCAT or GRE or something because they expect you to know this. But the 5' end of mature RNAs have this really funny nucleotide stuck on their ends. It's stuck on sort of in a backwards way because it's 5' to 5' triphosphate, not the usual 5' to 3'. And it has an unusual nucleotide in it where guanosine has been modified by the addition of a methyl group to cause the formation of 7-methylguanosine. Okay. Now, some of these other sugars, or in the beginning, or even the bases, can also be modified in other ways, but the cap is almost always there, and it's important for translation. But you don't have to know that. Okay, polyadenylation is next. Okay, that's the opposite end of the pre-mRNA. And it's important to realize that it's a bunch of A's. Well, they're nucleotides. We call them A's, but they're, of course, made from nucleotide triphosphates. But the important thing is they're non-templated. They're not copied from the DNA versus the rest of the pre-mRNA, which was copied from the DNA in a base pairing-like way. This occurs after a particular sequence, which is AAU, AAA, AAT, AAA in the DNA. And the actual polyadenylation occurs about 11 to 30 nucleotides after or downstream from this. So this polyadenylation signal is located within the 3' UTR after the translation stop codon. Polyadenylation doesn't, as you might think, just add on to whatever 3' end the RNA has. Actually involves cutting or chopping of the pre-mRNA so that the 3' end actually falls off and gets degraded. And then once you have this cleavage step, poly-A polymerase, not regular RNA polymerase, adds the adenosines to that newly exposed 3' end. Right, so here's a picture. We're finally to that point. We haven't got that far yet. We've taken off we've added our cap, we've had our 3' cleavage, and then we've added a poly-A tail. But we're not done yet because the introns still need to be removed. Splicing is actually a horrendously complicated process, and there's a lot of research going on into trying to understand how, when, and why particular introns are processed in the way that they are. So we're trivializing it by just going to the absolute bottom level of this must happen to get rid of the intron. There's a lot more to it than this. Okay, the first one is the GU-AG rule for intron borders. These nucleotides are generally, not always, but generally, the first and last within the intron. So the GU and AG end up getting spliced out with the intron. There's also a particular A within the intron close to the 3' AG, and that will form the branch point or lariat during splicing. The spliceosome is the protein RNA complex that actually carries out the splicing reaction, and it is made of protein and RNA. They are called SNRPs, which is to say small nuclear ribonucleoprotein particles, and the RNAs are called the uRNAs. So this is one of many cases where RNAs and proteins get together to form particular complexes which carry out biological functions. The ribosome, of course, is the more familiar case. Okay, so we're going to have to remove our introns. Here on the top we have the green is the exons, and they're going to be kept. Counterintuitive way of terminalizing that. Right, the first intron is shown. GU, AG are the first and last nucleotides of the intron, and again, those need to be removed. So here's the spliceosome, and in the process of actually doing the splicing reaction, very diagrammatically, that the spliceosome is composed of 5 different SNRPs, which are able to bring together the borders of the intron and get it cut and religated to produce the correctly spliced exons. So we're simply removing that intron in its precise way, dictated by sequences within the intron and proteins, etc., that are bound there. Okay, now clearly, changes in the DNA sequence can lead to incorrect splicing. For example, if those GU and AG splice junctions are mutated, we're not going to get correct splicing at that point, and that's going to mess up the protein big time. Okay, question. The GU-AG rule refers to locations of transcription start sites, splicing sites, ribosome binding sites, polyadenylation sites, transcription or termination sites. Okay, so the answer is B. Translation. People get confused between transcription and translation because of the words. Right. Translation is a decoding step where the nucleotide sequence information is converted to amino acid sequence. And the best analogy I can think of here is that translation involves translating or changing one language into another. In translation in biology, we are translating a nucleotide sequence language into an amino acid sequence language, whereas transcription is really meaning a copying. Like you can transcribe all you want without knowing what it means. You can just copy your nucleotides. But to translate is when you're creating a meaningful protein from nucleotide information by converting it to amino acid information. So it involves base pairing, but it's totally, completely different from that. But it does make an important point. The eukaryotic RNA shown, in general, produces a single protein. We don't have, like bacteria do, the ability to create multiple proteins from one single mRNA. In eukaryotes, translation occurs, it starts once on an RNA, and once it stops, that's the end of that. It does not reinitiate translation once it has stopped. Okay, that's important in terms of understanding what stop codons do, because if you have a stop codon early as a result of a mutation, the protein stops there at that point. There's no further translation from that particular RNA because it can't start up again, generally speaking. Okay, so here's our genetic code. We're going to learn how to read this. We'll have some practice reading this next time. Okay, and again, I want to show your globin RNA again, just to make a point about stop codons. Okay, so remember the open reading frame starts at nucleotide 51, continues to 494 rather, and it includes the AUG and UAA start and stop codons. Okay, so here we see it again. And if you were to sit down with this and actually patiently mark off groups of 3, you would see that you could create a protein with no stops in it, starting at that AUG and going all the way to the TAA, which is all the way down at nucleotide 491. Okay, so I can see this. I can do like I did before, sit down with my genetic code, read the codons from the genetic code. Okay. So the ORF ends when the first in-frame stop codon is encountered, and we've seen that already. Okay, my point is, actually, that don't think there are no stop codons within the open reading frame, because they are. They're all over the place. They're just not being read in frame. So I'm going to show you a few just by underlining them. They're everywhere. All right. So I underlined a whole bunch of them. Here's the TGA, 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 there's the TAA, etc. There are a lot more here that I'm not underlining, but this is just to make the point that there are stop codons scattered throughout this RNA sequence or cDNA sequence, whichever you choose to call it. Okay, but they don't count because they're not in frame with the AUG. So they're all over the place, but they're not being read because they're not in the groups of 3 that comprise the reading frame. Okay. So same sequence as before, just to make this point. Here's the first few nucleotides of the beta globin gene, and I'm reading it according to the rules of the genetic code in the usual way. But see, embedded here, we have a TGA stop codon, which is not being read as a stop because the nucleotides are split between those 2 sense codons that are in the right reading frame. Here's another one, a TGA, right, again, within the first 6 nucleotides of that, or first 6 amino acids of the protein. So those are stops, but they don't count because the ribosome is not encountering them in that way, and therefore they're not causing you any trouble. Now, this makes you think about frameshift mutations, however. Okay, so those stop codons make you think about what can cause disease. remembering the possibility of frame shift mutations. So if you add or delete a few nucleotides from the DNA, which ends up being part of the RNA, which ends up being translated as a protein, you're going to start bringing some of those out-of-frame stop codons into frame and causing the protein to stop at that point. So the reason why frame shifts are a problem is not really that it gives you the wrong protein, is that they give you a short protein because it's going to read out of frame until you get to a stop, which is going to be pretty soon, and that's going to stop the protein. So we will talk about mutations and diseases more next time.